Hey guys! Today I'm going to be helping you learn how to create and read Punnett Squares, and this should help you with the second part of your project. So you probably remember Punnett Squares from middle school. Usually this is an easier thing and kids love it because they remember it from middle school and it's easy to do. So Punnett Squares is just this diagram here, which is used to predict and compare possible outcomes from a genetic cross. So you put the father's alleles on one side of the Punnett square, the mother's alleles on the other side of the Punnett square. Remember, we have two alleles for every trait or every gene. And then you kind of bring them across. So the father's allele will come down, mother's allele will come across, and each of these boxes represents a 25% chance that that offspring will have that particular genotype. Okay, so let's do an example together here. So Gregor Mendel was a monk who found out these principles in his garden while he was pollinating his pea plants. So that's why a lot of these talk about Mendel, because Mendel is the person who started this. If you haven't done the Quizlet yet, which goes over some of the new vocabulary words, you should do that before we start. So you know words like homozygous and heterozygous, genotype. So homo, remember homo means same. Homozygous means two of the same alleles. So if Mendel crossed a plant with homozygous tall genes and a homozygous short genes, what will the genotypes for each plant be? So if the capital T is dominant for tall, right? The dominant is the stronger allele. Lowercase t is used to represent the short allele, the recessive allele. So homozygous tall pea plant would be two capital T's to represent the two dominant alleles. Again, homo means same. So if it's tall, it's going to have two of the same alleles. Homozygous short pea plant is going to have two small t's to represent the recessive allele. Okay, so now let's see what the possibilities for the offspring would look like from this cross. So how we do that is we put one parent's alleles on the top, other parent's alleles on the side, right? So we've got the homozygous tall on the top, homozygous short on the side. And all you do is you bring the top letter down and the letter on the side across to fill in each box. So each box should have two alleles, one directly above it, one directly coming from the side. And you just do the same thing for each box. So each box will have capital T, lowercase t. And if the capital T is the dominant allele for tall, all of these offspring from this cross will be tall, 100%. So you can see here we've got the tall pea plant, short pea plant, and all of their offspring will be tall, capital T, lowercase t. So now what happens when we cross two of those plants? And those are called heterozygous because they've got two different alleles, right? We've got the capital T and the lowercase t. So if we cross two of these plants, and remember from meiosis, each gamete, so each sex cell, and plants have sex cells too, each gamete only has one allele for each gene. So the offspring have one allele from mom, one allele from dad. So they're going to have two alleles for every gene. Okay, in this case, the gene we're talking about is the gene for plant height, right? Tall or short. Okay, so now let's look again. We'll set up our Punnett square. We've got one parent on the top, one parent on the side. Both are heterozygous. And if we do these out, we've got two capital T's here, capital lowercase t here, capital lowercase, and two lowercase. So what will the phenotypes be from these offspring? If you remember, phenotype means physical characteristics. So pause the video for a second and see if you can figure out what each of these plants will look like. Okay, remember capital T is dominant for tall, lowercase t is recessive for short. So if there's two capital T's, that offspring is going to be tall. Capital T, lowercase t will also be tall because the dominant trait for tallness is stronger than the recessive allele. Okay, so anytime there's a capital letter around, that's the one that takes over. The only time we see the recessive allele is here when there's two lowercase t's. That's the only time you can see that trait. So if each of these boxes represents 25%, this cross will 75% of the time will produce tall offspring. 25% of the time will produce short offspring. So you can also think of it as 3/4, right? 3/4 or 75% will be tall, 1/4 or 25% will be short. All right, my dog wanted to get in on this next part. So now those you've learned in middle school, those are for autosomal traits. So if in your project your gene was autosomal, so not on one of the sex chromosomes, that's what you're going to do. If your trait was sex-linked or X-linked, you have to do a little bit of a more complicated Punnett square. And I'm going to show you how to do that. So if you remember, for every human chromosome, we have a pair, right? So we've got two chromosomes, for each pair of chromosomes. Our sex chromosomes, though, females have an XX chromosome, so females have two X's. Males have an X and a Y. So males only have one X chromosome. So they're more likely to get X-linked traits because they only have one chance of getting the recessive allele. Whereas females can have two chances of getting a normal allele because they have two X's. So if the gene is recessive, they have a recessive allele and a dominant allele, which is normal, the normal one will take over, and females are usually just carriers of these traits. So the way you write out these Punnett squares are a little bit different. So you're always going to put an X and a Y, two X's to represent mom's genotype, and an X and a Y to represent dad's genotype. You're going to write the alleles up here at the top like an exponent. Okay, so for example, if you're looking at this key at the top of the slide, the capital B is representing a normal allele. This is for color blindness, so normal vision. The lowercase b is the recessive allele for color blindness. So a female whose X capital B, X lowercase b is a carrier. Her vision is fine because she has the dominant normal allele for vision. The only time a female will be colorblind if she has both recessive alleles, right? So female needs to have two lowercase b's to be colorblind. Whereas males, if you look here, a Y is a different chromosome. It's not homologous to the X, so it has different genes. So if we're looking at the color blindness gene, that's only on the X chromosome. Since males only have one X, they're either capital B, dominant, or lowercase b, recessive. Okay, so they either have normal vision or they have color blindness. They can't be a carrier because they only have one X chromosome. That makes them more likely to get these X-linked traits. So more males are colorblind than females. Male pattern baldness is another trait that's on the X chromosome, which is why you see more bald men than bald women, because they're more likely to get it. There's only got one X chromosome, so you either have it or you don't. Okay, so let's do one example together. Let me move my face out of the way here. So let's do an example of a couple. See how likely it is that a couple's offspring will be colorblind. So let's imagine a cross between a father who is colorblind and a mother who's a carrier, but she's not colorblind. So how likely will it be for them to have a daughter who's colorblind or a son who's colorblind? Why don't you pause the video for a second, see if you can figure out what the mother and father are, line them up on the side, and then if you can, try to figure out what the offspring will be. Pause the video and give it a try. Okay, so I'm going to give you the parent's genotype. So if the father's colorblind, he has his recessive lowercase b. Remember, nothing goes on the Y because the Y is not homologous to X. It doesn't have the same genes. The mother is on this side. She's got two X's because she's a female, and she's a carrier, which means she's heterozygous. So she's got a big B and a small b. Okay, now that you have that much, if you didn't do it all the way before. Try to fill in the Punnett square and see how likely it is that this couple will have a daughter who's colorblind or a son who's colorblind. Okay, so if you did it like this, you are correct. Now let's try and think about what the phenotypes for these individuals would be. Remember, color blindness is on the recessive allele, so the small letter. These two individuals are females. These are males, right? Females are XX. So these individuals here would be females who are carriers. So these girls would not be colorblind. These ones would be colorblind because they have two lowercase b's. So this couple has a 50% chance of having a daughter that's colorblind. Same for the boys. Only this box represents boys that will have the recessive allele for colorblindness. So 50% of the boys would also be colorblind in this example. Hopefully this helps. If you're still confused, I'm happy to do more examples during the Zoom meeting, or you can email me and we can talk about specifics to your project. Good luck!